Okay, so welcome back in another video tutorial. So in that video tutorial we are going to wrap up our discussion regarding RDT. Okay, so this is the last chapter that we need to know from recombinant DNA technology. So here we are going to discuss about the application of RDT. Okay, so we have discussed about various RDT technique for a long time. So today we are going to apply all of our knowledge, okay, so that we can use RTT for our benefit. Okay, so we are going to discuss here about lot of the technique where we will use our knowledge that we have acquired from different RDT chapters. So the first application that we are going to discuss, that already I have discussed earlier, that is insulin production. Okay, so first of all we need to know how insulin is naturally synthesized. Okay, so what is insulin? Insulin is a hormone that is produced by the islets of Langerhans cell of pancreas, okay, mainly the beta cell of islets of Langerhans. And what is the function of it? So it maintains the blood glucose level, as you can see. So remember one thing, insulin is first synthesized in the Prepro hormone form. Okay, that means inactive hormone form. Okay, so from that Prepro hormone, as you can focus on the structure of Prepro hormone, there is a signal peptide. Okay, and this is the B chain and this is the A chain, and it is joined together with the help of a C chain. Okay, and remember one thing, that insulin, the active form of insulin, consists of B and A chain only. That means during the pathway or during the conversion of Prepro hormone into an active form, C chain will be cut out in some point of the step. Okay, so the first step is the conversion of Prepro hormone into pro hormone. Okay, that means that pro hormone is structurally active but functionally inactive. So what is the conversion between Prepro hormone and pro hormone? So pro hormone, in the signal peptide will be cut away. Okay, as you can see. So the pro hormone lacks signal peptide, Prepro hormone have signal peptide. Okay, so from that pro hormone, then conversion of active heterodimeric hormone will occur with the help of an enzyme called pro hormone convertase 1, that will break the C chain. Okay, so as a result, A and B chain forms disulfide bond so that they can attach with themselves, as you can see, and as a result active heterodimeric functional insulin protein forms. Okay, so insulin is normally present in the pancreas as Prepro hormone. Okay, but whenever there is a need to control the blood glucose level, it activates itself by first converting themselves into pro hormone, and from pro hormone they will convert themselves into active heterodimeric protein. So now the fact is, if there is a situation when insulin is not well synthesized or not secreted from the islets of Langerhans of beta cell, what we should do? So we have to apply it from the outside, right? So what kind of drug we should apply? Whether we should apply a Prepro hormone insulin or whether we should apply a anti-heterodimeric form. So we will apply a anti-heterodimeric form, right, because it is the active form. So the goal of us will be to synthesize the anti-heterodimeric hormone artificially. Okay, then we will inject them inside the patient. So remember one thing, that insulin which is artificially produced are called recombinant insulin or humulin. Okay, that means insulin that is produced by human, that is why humulin. So as here we are wanting a expression of a protein, so what kind of vector we need to synthesize insulin? Yes, obviously expression vector. Okay, And remember one thing, the both A peptide and B peptide of insulin will be synthesized here with the help of two expression vector of the same type. Okay, and that means here two systems will run simultaneously. Okay, so as the normal property of a expression vector, it should have a promoter, it should have a transcription terminator, origin of replication, antibiotic resistant genes, and we will have to add insulin gene A subunit and B subunit just beside the highly expressive beta galactosidase gene. Okay, so what is the utility of attaching insulin gene A and insulin gene B just beside the beta galactosidase or just next to the beta galactosidase gene? So remember, this beta galactosidase gene here will act as a reporter gene, RG. So what is the function of that beta galactosidase gene or reporter gene here? So as we have mentioned that beta galactosidase gene is a highly expressed gene, so it will ensure whether promoter is active or not. Okay, and later it will also help us to purify insulin gene A and insulin gene B from the mixture of protein, right? Because here we are willing to transform those recombinant vector inside a Equalized, right? So after the expression process, we have to purify them from the total protein samples, right? So here will be the mixture of protein coming from Equalized and from those recombinant vector also. So here beta galactosidase gene will help us to purify our insulin gene E subunit and insulin gene B subunit from those mixture of protein. Okay, with the help of a process called affinity chromatography, right? So that is why we have inserted insulin A gene and B gene next to beta galactosidase gene. Then our next idea will be simply insert them inside a Equalized, okay, by a suitable transformation procedure. So here we are using heat shock calcium chloride method, right, which is specific for Equalized, and then we will select them with the help of a antibiotic resistance gene, right, that we have incorporated. Okay, now we will incubate them. Okay, and during this incubation, the expression for formation of protein, that is a fusion protein, in one set we will have insulin gene A protein along with beta galactosidase, and in another set we will have insulin B with beta galactosidase. Okay, now our goal will be to purify the fusion protein from the remaining protein. Okay, so here beta galactosidase will help us, I have already mentioned. Then our job will be to remove that beta galactosidase from the A subunit to B subunit. So it is extremely easy. We will have to add cyanogen bromide. Okay, so in presence of cyanogen bromide, A subunit and B subunit will be removed from the beta galactosidase. Okay, so cyanogen bromide will simply break the methionine that separates the beta galactosidase with the A subunit and B subunit. Now we will have the A subunit and B subunit, and then we will simply attach them by keeping those A subunit and B subunit in appropriate environment, so that the disulfide bond will form between A subunit and B subunit, and it leads to the formation of active heterodimeric insulin, okay, that we can use as a drug. Okay, and what is the utility of that process? First of all, before the development of RTT, insulin is artificially synthesized in a cell line. Okay, and as a result the cost of insulin become very high. Okay, and it cannot be affordable for normal people. As a result, the demand of insulin become very low. But recombinant DNA technology help us to synthesize those insulin in simply the prokaryotic systems that is easily available, right? So that is why the insulin, that is this one, become affordable for normal people. As a result, the demand of insulin in the world of medicine become very high. So that is the utility of recombinant DNA technology in production of insulin. Now the next one. So here we are going to discuss about how recombinant DNA technology help us to produce human growth hormone. Okay. So first of all, we need to know what is human growth hormone. Human growth hormone is a hormone that is secreted by pituitary gland. Okay, and it will help you to form the normal body growth and development. Okay, so it will control the normal body growth and development. Along with it will also involved in development of muscle, bone, and lengthening of height, etc. are all regulated by human growth hormone, right? So muscle, bone, and lengthening of the height all are regulated by human growth hormone that is secreted from where? Pituitary gland. So low production of human growth hormone result in the dwarfism, and high production of human growth hormone result in the formation of giant structure, right? And that is we see in case of Great Khali. So for him, the human growth hormone is secreted at a very large amount. That is why he have a very giant structure. So what are the solution that was used before the development of recombinant DNA technology to fulfill the scarcity of human growth hormone in those people in which the human growth hormone is not well synthesized, right? So for those peoples, the growth hormone from diseased human were treated, okay, especially from the age 1 of those children who lacks growth hormone, okay. And remarkable example of growth hormone deficiency is so a remarkable example of growth hormone deficiency was observed in case of so children of age 1 were treated with growth hormone from diseased human. So this therapy continues for 8 years, but there is a risk of transmission of the disease, okay. So that means, because just try to think about this, that here we are injecting growth hormone from diseased human, right, which those who have already expired, right. So there can be chance of transmission of disease, right, and here we are constantly giving the therapy to that children from age 1 to age 8, right. So as a result, the cost of those growth hormone therapy becomes very high. So it is not affordable to normal people. So here now human recombinant DNA technology takes the charge or takes the responsibility to make it affordable to normal people, okay. So human growth hormone is a hormone that is secreted by pituitary gland of human, okay, that involved in body growth and development, okay, and lengthening of the height. If human growth hormone is produced in low amount, it will result in dwarfism, and if it is produced in high amount, it will result in formation of giant structure. So before the development of human growth hormone, the growth hormone was injected, okay, from diseased human, okay. In case of child whose age is 1, okay, from age 1 to age 8, a therapy is constantly given where simply growth hormone from diseased human was injected, okay, and there will be a chance of risk of transmission of the disease is also there, and also there is a chance that the cost of the therapy is very high, so it is not affordable to normal people. So now we are going to discuss about how RDT makes it easier to provide growth hormone to those people who have scarcity of growth hormone. Okay, so first of all, we need to study about the sequence of human growth hormone coding DNA. Okay, so how we can do this? So from the mRNA that is synthesizing human growth hormone, from those RNA with the help of reverse transcriptase we are synthesizing cDNA. Okay, so now by analyzing cDNA we find that there is a signal sequence which is a mammalian signal sequence, and the red marked portion is the main sequence that actually converted into human growth hormone protein. Okay, this red mark portion, and there is another small sequence where there is a cut site for HIN3. So this is the normal sequence of human growth hormone cDNA. Now the question, there will be a question that why we do not perform the cDNA technique in the case of insulin. So remember, by observing the DNA sequence in case of insulin, we find that there is no exons, no introns. Okay, so the use of cDNA is of no meaning in case of insulin, right? Because why we are using cDNA, that we have discussed in case of our cDNA library, to exclude the introns, right? That is the main goal of using cDNA. But in case of human growth hormone, it is find that it is observed that human growth hormone DNA contains introns, right? So we need to exclude them to produce a DNA molecule that will only contain the exons, right, or only contain the coding genes. So that is the reason why in case of insulin we do not perform any kind of cDNA, but in case of human growth hormone we have to synthesize cDNA, because human growth hormone, the normal DNA sequence of human growth hormone possesses our introns, right? So what will be the first step? So our first goal will be to remove the signal sequence. Why? Because remember, we only need the red marked portion, right? That is the main coding sequence for the human growth hormone. So as we can see that here is a cut site for EcoR1, so we will simply cut them with the help of EcoR1. So as a result, a codon for amino acid 1 to 24 will be removed. Okay, now the first thing is that scientists tried to only insert that part into a vector and then transform them into E. coli, but they observed that in such cases the protein become not well folded and they become simply non-functional, right? So that is why they understand that yes, we also need the codon for 1 to 24 amino acid, right? So now what to do? So here we are using simply a synthetic oligonucleotide that will provide those codons for 1 to 24 amino acid. Okay, but it is not that the oligonucleotides will be exactly same that we have removed. No, because we all know that various codons there are that will code for the same amino acid, right? So we can use any kind of oligonucleotide that will give us that 1 to 24 amino acid. Okay, so we have simply ligated this with the remaining portion. So here as you can see that there is a eco on one side, so most of the cases it will be a staggered cut. Okay, so that the ligation will be very well and easier, and we will ligate them with the help of T4 DNA ligase, right? So after ligation we will have something like that. So it will have a HIN3 cut side. Why we have incorporated a HIN3 cut side we are going to discuss, and there will be a cut side for EcoR1, and at the end there will be also a HIN3 cut side. So I hope you can understand already that why we have put two HIN3 cut side into end of the DNA. Yes, obviously, because we are going to ligate them in the vector with the help of HIN3. Okay, so here there is the first codon, that is So that is the first amino acid codon, that is for the methionine. Okay, now we will cleave both of them with the help of hin tree, and ligation of our gene of interest inside a vector will occur. And as here we are again looking for expression, so yes, there will be a promoter, then there will be initiated methionine and growth hormone coding sequence, and then ampicillin resistant gene. There should also be a origin of replication and transcription terminator. Okay, so now we will transform them simply inside a E. coli with appropriate transformation method, and then growth hormone accumulation inside the bacterial cell occur. Okay, so once satisfied amount of growth hormone is synthesized inside the bacteria, we have to simply lyse them, and we will isolate the growth hormone from the mixture of the protein, right? So that is the normal procedure, but there is a problem. What kind of problem it is? So remember, in that case the crowding of unnecessary protein is too much high. Okay, that means the crowding of E. coli protein is too much high. As a result, isolation of growth hormone from those heavily crowded unnecessary E. coli protein become a very mammoth task. So as a result, we need to develop something to simply purify them, right? Because if we have to perform a mammoth task to purify human growth hormone from those unnecessary E. coli, so it will simply reflect in its cost, right? Because the cost will be higher. So that is why scientists need to think about it again, and they develop another procedure so that they can easily isolate human growth hormone from those crowded E. coli proteins. Okay, so what they do, so let's go back to the second step where we are removing the codon for amino acid 1 to 24 and replacing them with synthetic oligonucleotide. So they suggested that instead of using any kind of oligonucleotide that will only complete the need of 1 to 24 amino acid, let's put a bacterial signal peptide. Okay, that is a signal peptide for E. coli secretory protein. Okay, remember this, a bacterial signal peptide for E. coli secretory protein. Okay, and the rest of the procedure remains the same. So what will be the utility of that insertion of a bacterial signal sequence? So remember, here our initiator methionine will be here at the starting of bacterial signal sequence. Okay, so now the question is, what is the utility of keeping a bacterial signal sequence here? So here the idea is, as it is a signal sequence for secretory protein, so E. coli will recognize that signal sequence and secrete our recombinant protein in the periplasmic space. Okay, because don't forget that E. coli is a gram negative bacteria, so it will have a periplasmic space, right? So we will simply secreting the periplasmic space, so the recombinant protein will simply released in the periplasmic place, right? By due to the presence of bacterial signal sequence that is for E. coli secretory protein, so that E. coli can recognize that yes, I have to secrete that protein in the periplasmic space. Okay, so now the question is, what is the utility? And remember, during the periplasmic during its entry in the periplasmic place, the periplasmic protease will simply cleave off the signal peptide. Now we will simply release the HGH with the help of periplasmic shock. So now what is the utility of keeping a bacterial signal sequence for the E. coli secretory protein? So here the utility is, the protein that is present along with the HGH in the periplasmic space is much lower in amount than the protein in case of the when we are trying to purify HGH from a total mixture of protein. Okay, it is obvious, right? Because the amount of E. coli protein in periplasmic phase should be lower than the amount of other E. coli protein, that is in case of the total protein, right? When we are simply lysing the E. coli and try to isolate our periplasmic protein, our human growth hormone from the total amount of protein, right? So as a result, the purification process become easier, so it will affect in the cost, and the cost become lower, right? Because here we do not need too much work, we do not need to perform too much work to isolate our human growth hormone. Remember this, in such kind of human growth hormone is known as methionine less human growth hormone, okay? Although it do not cause too much difference with the methionine containing human growth hormone, okay? So there can be a difference between what is the methionine less human growth hormone and methionine containing human growth hormone. So the basic difference is methionine less human growth hormone containing a bacterial signal sequence, right? That is especially of Equalized secretory protein, okay? So here, as initiator methionine is in the starting point of bacterial signal sequence, that will cleaved off by the periplasmic protease, as we can see, so it will lacks methionine, okay? So and in case of methionine containing human growth hormone, there is no bacterial signal sequence, right? And here, as there we are not secreting the protein in the periplasmic place, so there is no chance of cleaving of initiated methionine. That is why the methionine containing human growth hormone contains initiated methionine. And you can also remember that in case of methionine less human growth hormone, it do not need too much mammoth task for purification of human growth hormone, but in case of the methionine containing human growth hormone, the purification becomes difficult. So that is the concept behind the synthesis of insulin, the synthesis of human growth hormone with the help of recombinant DNA technology. So human growth hormone is a hormone secreted by pituitary gland that will play a important role in the development of muscle, bone and lengthening of height, okay? So for those person who have scarcity in human growth hormone, from the age 1 to age 8, age therapy was performed, where we are simply inserting the growth hormone from diseased human, right? So in such cases, there is a chance of disease transmission, and also the cost of those therapy is very high, right? So now recombinant DNA technology came into the picture, and it makes, or it provides the way to produce human growth hormone artificially, right? And in a very cost effective manner. As a result, the drug for human growth hormone become affordable, and it gains a high demand in the medical world. Now another application we are going to discuss, that is on the agricultural biotechnology, okay? So here our discussion will be on production of antisense molecule. So I hope you know about the transgenic tomato, right? So what is the main idea of transgenic tomato? So remember, in case of tomato plant, there is a enzyme present that is called PG enzyme. The full form of PG enzyme is polygalacturonase, okay? So this polygalacturonase have the ability to cause rotting of tomato, okay? So within some days, okay, near about 2 to 3 days. As a result, it causes a huge loss in the economy, right? Because most of the tomato become rotten. As a result, it cannot be sold properly. So now again RDT come into the picture. So here the idea is to synthesize a single stranded complementary RNA molecule, okay? So remember, the RNA molecule that can be translated into protein are known as positive sense RNA molecule. Okay, and the complementary of that RNA molecule is simply referred to as negative sense RNA molecule. Okay, and remember, from negative sense RNA molecule there is no translation of protein. That is why in case of HIV virus replication we have seen that from negative sense RNA the first job of HIV will be to convert it into a cDNA. Okay, from that they will convert them into positive sense mRNA, right? So that is the reason why. So this negative sense RNA will not going to produce any kind of protein. So here our goal will be to produce a negative sense RNA of the complementary of the positive sense RNA that will translate the polygalacturonase. Okay. So what will be the utility of that procedure? So utility of that procedure is when we will add those antisense molecule, it will simply interact with the positive sense RNA molecule, and as a result a complementary double-stranded RNA formation will occur. Okay, and as a result the polygalacturonase is no longer synthesized, right? Because we all know that from single-stranded mRNA molecule synthesis of protein will occur, but here we are forming a complementary double-stranded RNA molecule. So here it will be RNA. Okay, RNA molecule. So as a result no synthesis of polygalacturonase occurs, and it will gives to the formation of ripe tomato where no rotting is observed for about 3 weeks. Okay, so what is the procedure? So here remember we are using the same procedure that we have discussed in the chapter 4, that is the Agrobacterium tumefaciens mediated gene transfer. That is a standard method for formation of any kind of transgenic plant. Okay, so I think you know that in case of Agrobacterium tumefaciens there will be a left border and right border. Okay, that is the flanking sequences that flanked the tDNA, right? And here our virgin will produce the protein that will make a nick in both two sides and yield a single-stranded copy of the tDNA. Okay, and now a protein of virgin will guide that single-stranded tDNA copy inside the plant cell, right? So in our expression vector presence of left border and right border is necessary. Okay, so that the protein from the virgin will make cut. Okay, along with the cloning site, here multiple cloning site is preferable, and a NP2 gene for kanamycin resistant selection, it is also necessary. We all know, we have also discussed this in case of our normal transgenic plant production via Agrobacterium tumefaciens. Okay, where here we have also incorporated the kanamycin resistant, and a bacterial origin of replication will be there, and marker for selection of bacteria will be there. Okay, so remember one thing here that you need to remember, although here we are also producing mRNA, but here we are not need any kind of promoter. I hope you can understand why, because it will integrate with the plant cell nucleus, and there it will transcribe into mRNA, right? So why we need expression vector? So here we can do our job with the help of a normal cloning vector too. Okay, so now this is our gene of interest. Okay, so although here we have incorporated a strong promoter, but I think but it is of no use, because try to think about this, the virgin will simply cut the left border portion and right border portion and simply yield the single-stranded DNA copy and integrate that single-stranded DNA copy in the plant cell nucleus, right? But there is a problem. what problem it is? So remember, here we will also going to use the disarmed TI plasmid, right? So in that disarmed TI plasmid, there will be field region, which is activated field region, but there will be no promoter, right? So we have to incorporate a promoter for the synthesis of that feed gene into protein, right? Otherwise the cleave of left border and right border will not occur, right? That is why we have to incorporate a promoter. Okay, I hope you have understood. So there will be a fragment from 5' end of the cDNA encoding polygalactouronase clone in antisense orientation, that is the transgene. So we have ligated it into the cloning site. Now we will simply transform it into E. coli, and transformed the cell are selected. Okay. So now this is inside the E. coli. Okay. So from that E. coli we will simply isolate that recombinant plasmid, and we will simply insert them into a Agarobacterium tumefaciens bacteria, where we have a helper plasmid, that is nothing but the disarmed TI plasmid containing virulence region. Okay, and there will be a formation of co-integrative plasmid, as we have discussed in case of our transgenic DNA plant formation. Okay, so vir gene will synthesize vir protein. Okay, this vir protein will simply help in yielding the single stranded DNA copy of the gene of interest by cleaving the left border and the right border. Okay, now there can be another thing that we can do, that process that I have discussed with you, that is the normal transgenic plant forming procedure, but here we can perform another technique too. We will do not go for any kind of co-integrative plasmid formation. Okay, so here it will simply play the role of helper plasmid, and this CAMB strong promoter will help that virulence gene to produce the vir region, and here the vir region will have a high affinity, okay, for the left border and the right border, and they will cleave simply left border and right border. Okay, leads to the formation of a single stranded DNA which will contain our gene of interest. Okay, so this can be also done. That means here we are not going to use any kind of co-integrative plasmid. Here helper plasmid simply synthesize the virulent protein for us, and this virulent protein will simply cleave the left border and right border and generate a single stranded part containing gene of interest, and now a protein of vir gene will guide that single stranded DNA containing gene of interest inside the plant cell nucleus via a process resembled to conjugation. Okay, so as a result, a plant cell containing gene of interest inside its nucleus will form, that will regenerate the plant, and as a result transgenic tomato plant will produce. Okay, that will remain ripe for near about 3 to 4 weeks. Okay, as a result of complementary RNA formation. Okay, so now we are going to discuss about the Bt toxin formation. Okay, so what is the Bt cotton? What is the idea of Bt cotton? Although we have discussed about Bt cotton earlier, so here the Bt means Bacillus thuringiensis. Okay, so it is a bacteria that produce a toxin from the cry gene. It is a gene that is present inside the Bacillus thuringiensis, but although the toxin is in inactive form. Okay, so this toxin will become activated inside the gut of the insect, midgut of the insect, due to the alkaline pH of the insect midgut that is preferable for the activation of that Bt toxin. Okay, and as a result, those Bt toxin will simply bind with the midgut in the insect midgut cell and activate cell death pathway and also creates various pores inside the insect midgut, and a result lysis of the cell occur. Okay, so what is the actual idea here? So here we will simply isolate that the gene that is responsible for the formation of Bt toxin, okay, which is in inactive form, and luckily this inactive form remain inactive in the gut of human, and it is also remain inactive in case of plant, right? So with the help of Agrobacterium tumefaciens, if we insert those cry genes inside the plant, it will not cause any problem, okay? But when insects, especially bollworm in case of cotton, are going to eat those kind of transgenic plant, it will also consume too much amount of toxin. Those toxin will simply become activated in the insect midgut. Okay, so remember, in the bacteria, in the plant, in the human, the toxin remains in crystallized form. Okay, but insect midgut will provide a appropriate alkaline pH that will convert that crystallized toxin into solubilized toxin, and as a result the toxin become activated. Okay, and now those activated toxin will bind with the insect midgut, activate a cell death pathway, and also causes pores in the midgut, and as a result the cell lysis will occur and cell dies of starvation. So here we are not going to discuss about the process because we have discussed about the process earlier. The simple thing that we have done in case of the transgenic tomato formation, or the simple thing that we have done in the synthesis of any kind of transgenic plant, right? That is the Agrobacterium tumefaciens mediated gene transfer. So now another utility, that is gene therapy using adenovirus vector. Okay, so what is the term gene therapy? So here artificially we are simply incorporating a gene inside our cell of interest. Okay, as a result the gene will be expressed itself inside the cell, and it can have the ability to solve any kind of genetical disease. Okay, so gene therapy is a idea or is a treatment for the genetic disease, okay, where we are simply incorporating new gene, okay, that help us to survive against the genetical disease. Okay, so here we are going to discuss about the adenoviral mediated gene therapy. So the first goal is to simply insert the new gene in between the viral DNA. Okay, then we will insert the modified DNA into the vector. Here the vector is nothing but the virus. Okay, now vector will be packaged in vesicle, that means receptor mediated endocytosis facilitate entry inside the cell or inside the host. It will break down, the vesicle will break down and releasing the vector. Now this vector will interact with the surface receptor present on the cell of interest and inject the nucleus, and here the nucleus is inject the nucleic acid that it carries. But remember, here the nucleic acid is not its gene but our gene of interest, right? So cells start making protein using those new genes. Okay, so here with the help of recombinant DNA technology, we are using virus to make a target specific gene therapy, because remember virus are very target specific, right? So that is the reason we can use virus to provide gene therapy. And remember one thing, what is gene therapy? Gene therapy is simply a procedure, okay, by which we can cure any kind of genetic disease. Okay, so now we are going to discuss about vaccine formation. We have hepatitis B vaccine formation with the help of recombinant DNA technology. So what is hepatitis B? So hepatitis B is a very infectious illness of the liver caused by hepatitis B virus. Okay, so remember, in hepatitis B virus there are three surface marker present. Okay, one is hepatitis B surface antigen, one is hepatitis B envelope antigen, and hepatitis B core antigen. Okay, so we are simply using hepatitis B surface antigen, because it is the main immunostimulation part. Okay, surface marker. So what will be our goal? Our goal will be to simply amplify the hepatitis B surface antigen with the help of any kind of expression vector, and then we will simply add them inside in the human, so that the human immune system will produce antibody against the hepatitis B surface antigen, or we can also produce the antibody against another animal. Okay, now by simply isolating those antibodies, we will ready-madely provide immunoglobulin to a human. Okay, that is the different approach that we can perform. So here our discussion will be how we can produce hepatitis B virus vaccine. Okay, so let's discuss. So hepatitis B virus vaccine. So remember, as it is a eukaryotic gene, we need eukaryotic host for their expression. Okay, although we are expressing various eukaryotic gene inside the prokaryote, but we cannot do this in case of hepatitis B surface antigen. Otherwise, non-functional hepatitis B surface antigen will produce, which will of no use. So remember, as you can see that we have a origin for bacteria and origin of yeast, a tetracycline resistant gene, a LEU2 resistant, a LEU2 gene present here, and a yeast promoter here also. Okay, so what we can conclude, that here we are using a shuttle vector, right? A vector that can shuttle between yeast and E. coli. So this will be obviously our MCS site. So here we are incorporating the hepatitis B surface antigen by simply taking the hepatitis B surface antigen from gene library. Okay, we have a gene library for hepatitis B. From that we can simply isolate hepatitis B surface antigen by using colony hybridization or by using colony PCR. This is totally depends on you what procedure we can use. Now we will incorporate those hepatitis B surface antigen here. Now we will transform them inside a yeast. Okay, and